Okay, so for the past couple days, I have spent 18 hours creating the best AP Bio notes for your use on the AP tests. There will be a lot of these videos coming out, one per unit. So if you like this one, I'd really recommend subscribing so you know when the next one comes out. But just to prove to you how good this stuff is, we're going to go through this kind of stuff. First, we're going to start with biochem and macromolecules. Before we get into like any real bio stuff, we do have to go over some basic chemistry. So you have to know what an ionic bond, which is a bond where there's an attraction between metals and nonmetals, and it goes away in water. So this is like your salt, right? You have your metal, which is sodium, and your nonmetal, which is chloride, binds together, sodium chloride, salt. Covalent bonds, which is when there's an attraction between nonmetals who share electrons in the outer shell, and this like stays in water. It doesn't break apart. A example of a covalent bond would be water, but also glucose, oxygen gas, carbon dioxide, carbon monoxide, methane, etc. And speaking of water, let's talk about hydrogen bonds. So when your molecules are sharing electrons, some molecules or some atoms will get more electrons than the other, right? Because they're just much bigger, and the electrons will gravitate to that atom more often. So as you can see with this one, the oxygen in dihydrogen monoxide or water is slightly negative, while the hydrogen atoms are slightly positive because they are much lighter than the water molecules, and therefore losing that attraction, or losing that electron, making them more positive. And this is just a thing that you might have learned from physics or, batteries, basically, magnets, stuff like that. But a negative charge and a positive charge attract each other. So this hydrogen is attracted to this oxygen, and this hydrogen attached to that oxygen is attached to this oxygen, and it just keeps on going round and round. And this gives water a bunch of properties. For example, it's highly polar. It has cohesion, where it can attach to itself. If you've ever seen water droplets on your window and they're all like of varying sizes, that is from that. Surface tension, which is when water is more cohesive because the top shares less hydrogen charge. Basically because it doesn't have to share the water charge with the air. It can actually just kind of bubble over, and that's how you get your surface tension. Adhesion, when water sticks to charged items, right, or just polar things. This is why you have to measure at the meniscus when you have your beakers, right? As well as capillary action. Oh, that's the same thing. When it climbs up to the sides of things, right? So adhesion could be mostly anything. For example, let's say you had a kickboard in swimming. You had your kickboard. That kickboard will still have some water on it, even if you flip it upside down and move it all around, because it's just going to be attracted generally to the kickboard. It has a high specific heat, which means that the water can absorb a lot of energy before changing, not shape, form, changing stages of matter. So it'll take a lot more energy to get water to turn into steam. And this is important because it allows vaporization to occur, right? So your body releases this water, and it cools down your skin in order to heat up this water to make it evaporate. Now let's talk really briefly about what is the difference between a polar molecule and a nonpolar molecule. So whenever a molecule has that thing where like parts of it are negative and parts of it are positive. That is a polar molecule. It is hydrophilic, which means it loves water, okay? It can mix with it at any time. However, nonpolar molecules are ones where there is no like dipole or like sharing or uneven sharing of electrons. So that's more like oil. So when people say, oh, these two mix like water and oil, that's because water and oil literally physically cannot mix because it is hydrophobic and water is hydrophilic. Or yeah, because it obviously loves itself. Macromolecules. So macromolecules are your proteins, your carbs, your lipids, your nucleic acids. We're going to be talking about nucleic acids later in this like whole thing with AP Bio, but we're still gonna talk about proteins, carbs, and lipids. These 4 things are required by every living organism, and they're made of monomers, which usually become polymers. Okay, and these polymers are relatively large. First one we're going to talk about is carbohydrates. So carbohydrates are used for 3 things mostly, which is immediate energy, structure, and stored energy. It's made out of carbon, hydrogen, oxygen, specifically this formula, CH2O times N. So if you're thinking glucose at C6H12O6, your N would be 6. Right, and that's not counting like dehydration synthesis or anything, which we'll get to into a second. Monosaccharides are just one carbohydrate, which is just like the building blocks. So if you've ever heard of glucose, galactose, fructose, that is a monosaccharide, and it is used for instant energy just like that. A disaccharide is used for immediate energy too, yes, but it's also used more as like a short-term energy storage. You have lactose, which is glucose plus galactose, sucrose, which is glucose plus fructose, and maltose, which is glucose plus glucose. I doubt on your AP test it's going to ask you that kind of thing, and if it does, it's going to be a multiple choice question. But for the sake of curiosity, I would recommend learning this. But the more important thing is the whole deal of polysaccharides. So polysaccharides are made of tens to thousands of monosaccharides, and they can take all these like different shapes. For example, cellulose and chitin or chitin are basically straight lines of glucose. Cellulose is in plants, and chitin is in animals as well as fungi. Right, and that's what gives it its structure. So if you ever seen like an exoskeleton, or you ever hear that crunch when you're eating like lettuce or any other kind of vegetable, it is because of the cellulose. Now you have starch in plants, which is the thing that's found in potatoes, and it's a loose coil of glucose, and that's used for like long-term energy storage, way longer than any kind of disaccharide. That's why potatoes have a lot of them. That's that kind of thing. Glycogen is similar to that, except it's for animals. When you have branching loose strands of glucose used for long-term energy storage. So I think one way to think about it is the glycogen is branched out while the starch is like a staircase. That's how I'd separate the two. I mentioned dehydration synthesis, and now we're going to talk about that. Dehydration synthesis is when you take out one H2O molecule from 2 monomers, right? And that's going to be a hydroxide from one side and a hydrogen from the other side. When you take those 2 away, they will combine into one single molecule. So if you had 2 monomers, that would become, I don't know if there's like a donomer, but if you had 2 monosaccharides, that would become a disaccharide. And then you could do the same thing with another one. So if you had those 2 combined molecules and you did another dehydration synthesis, you could add another one, and then another one, and then another one. And that's the whole idea of dehydration synthesis. You're just taking out of water and you're putting these 2 together. Now the complete opposite of that is hydrolysis or hydrolysis, where you use one H2O molecule to basically break apart a polymer into monomers. Now let's talk about lipids right over here. Lipids, aka fats, steroids, phospholipids, oil, or wax, are made out of carbon, hydrogen, and oxygen. And if you're a phospholipid, you also have a phosphate in you. It is used for long-term energy storage, membranes, insulation, and cushioning. Fats and oils are triglycerides, which means that they're made of one glycerol and 3 fatty acid chains. And these fats, what differentiates them, you have your saturated fat, your trans fat, your unsaturated fat. You can tell the difference between these based off their fatty acid chains. So a saturated fat has only single bonds, right? So you have straight lines of hydrogen happening, right? Like this, so you have or carbon. So you have carbon, then carbon, then carbon, then carbon, just like that. And on each side, you have a hydrogen, and a hydrogen, and a hydrogen, and a hydrogen, right? That's what a saturated fatty acid chain would look like. However, with unsaturated, it would have at least one double bond between your carbons. So you'd have a double bond right here, and you'd have one hydrogen here, carbon, carbon, just like that. And that would have less structure to it compared to a saturated fat. So saturated fats at room temperature are more solid, while unsaturated fats are more liquid at room temperature. And then you have your trans fats, which are like these artificial fats that you'll find in your donuts and stuff. And instead of having a double bond like the one I just showed with unsaturated fats, let me see if I can get a better pencil. Unlike the one in unsaturated fats that I just showed, it will be one goes up and one goes down, instead of unsaturated, which is like this. Okay, that's basically it for lipids. Let's talk about proteins. Okay, proteins. So proteins are used for structure, enzymes, which we will talk about later, and a last resort of energy, as well as transport, communications, and signaling. But the main ones are structure, enzymes, and last resort of energy. So proteins, instead of being made of monosaccharides, glycerides, or fatty acid chains, are made of amino acids, right? This right here is what an amino acid looks like. Let me try zooming in. Oh, just let me move that over. There we go. So, an amino acid has a carbon right in the middle, a hydrogen right at the top, an amino group to the left, and a carboxyl group to the right. At the bottom, you will have an R side group or an R group. A side group or an R group. Now, there are 20 naturally occurring amino acids, each with their own unique R group. And these amino acid, special R groups are what give amino acids their very, differing roles, purposes, and stuff. So amino acids bond with dehydration synthesis as well to make a dipeptide. So when you have one peptide, another peptide, or one amino acid, another amino acid together, that makes a dipeptide. This is called a peptide bond. Pretty simple. There are 4 levels to protein structure. First, you have primary, which is just a chain of amino acids held together by peptide bonds. And the second is when it twists into an alpha helix or a B-plated sheet because of the hydrogen bonds, right? So you have hydrogens here, oxygens here, and the way that everything is structured could lead to a alpha helix, which is like a staircase-looking thing, and a B-plated sheet, which looks like a book. I would actually recommend taking a look at this, researching that to get more insight. Next is tertiary. It is when a protein globs up because of R group interactions or a sulfur molecule. And then quaternary is when the shape made up by multiple proteins to make a functioning protein. And the globbing up that's mentioned in the tertiary structure is talked about right here. There you go. So globbing rules for tertiary structure. The strongest interaction between R groups containing sulfur make disulfide bridges. So this basically folds your protein in half wherever these two things meet. They're like magnets, right? They're super, super strongly attracted to each other. Polar R groups orient outside while non-polar orient inside. Realistically, you're not going to need to know this, but just in case, you always want to be well prepared. Denaturation or denaturation. You want to know this for sure. It's when a molecule or a protein falls apart because of external thermal, kinetic, or other UV energy or like pH stuff overpowers the energy holding the protein together, right? So think about, this could be attributed to humans too. Let's say a human, all he has to do is press a button. That's his one job. And he's pretty efficient at it. But if you lower the temperature, he's going to get cold, and it's going to be harder for him to press the button until you have a human icicle. Or pH. If you have a man pressing a button, he's pretty efficient at it. If you dump hydrochloric acid on him, he's going to not be as efficient at it because he's going to be dead. So yeah, that's just a way to think about denaturation. It's things stop working to a specific level. Metabolism. Metabolism is the sum total of reactions in a cell or organism. Some like terms that might show up on your test, like pop quiz style, is catabolism, which is the breakdown of molecules to release energy, and anabolism, which is binding of molecules thanks to energy. So the way I want to think about it is catabolism is like catastrophe, right? Like it is blowing up. Well, anabolism is like anabolic steroids, okay? Which build muscle. So clearly it's building things. And when it builds something, it's thanks to energy from somewhere. Reactions are quantified with Gibbs free energy, aka delta G, with this equation right here. And this is how you basically find out metabolism. So you have delta G is equal to the change in temperature minus the temperature in Kelvin times the change in entropy or disorder. This stuff will be given to you on your formula sheet, I believe, as well as like all the things that you need to know, like what's this, what's this, what's this. It's going to be given on the question somewhere. So you don't really have to worry about that one. And most likely that's not really going to be a multiple choice question. There's going to be more to it. Yeah, okay, that's actually everything for macromolecules and biochem. So good job. I will see you guys in the next video, and good luck on your tests.